Okay, so Chapter 5, Section 3, Active Transport. So active transport moves solutes against their concentration gradients. This is opposite of what happens with passive transport. So cells maintain an internal environment with a different composition than the outside environment. This requires work, energy from ATP is needed to move substances against their concentration gradients. This is called active transport. Specific carrier proteins move substances in only one direction, either into or out of the cell. So, for simple diffusion, there's no energy needed. It's down the concentration gradient. There's no membrane protein required, and there's no specificity, other than it can't be polar or big. Facilitated diffusion. This is through a channel or carrier protein. Cellular energy required? No. The driving force is the concentration gradient again. It's going down the concentration gradient. Membrane protein required? Yes. Specificity? Yes. It only works for one. Okay, active transport. It requires energy, usually in the form of ATP. The driving force, ATP hydrolysis. So this is ATP going through hydrolysis, which is the breaking apart of a polymer into monomers. So here we have our adenosine triphosphate. So one of the phosphate groups breaks off through hydrolysis. So water goes in and a phosphate leaves. So this releases energy for the moving of the particle. So membrane proteins are required, and it only works for one type. So two types of active transport. There's primary active transport, which involves direct hydrolysis of ATP for energy. There's secondary active transport, which uses the energy from an ion concentration gradient or an electrical gradient. The gradients are established by primary active transport. So primary active transport uses ATP, secondary active transport uses ion concentration gradients. So the sodium-potassium pump is an integral membrane protein that pumps sodium out of a cell and potassium in. One molecule of ATP moves 2 potassium and 3 sodium ions. So here we have outside of the cell, there is a high sodium and low potassium concentration. So here's the pump. Potassium, sodium goes in to the 3 open holes. It then gets energized by ATP, and it moves those sodium out of the cell. This changes the conformation of the protein, which opens up these squares so that potassium can go in, and closes up these so that nothing can go in. This then allows potassium to go through, releasing the phosphate group and reopening these for sodium to reattach to. So this gives you a lot of potassium in here and a lot of sodium out here. So secondary active transport uses energy that is regained by letting ions move across the membrane with their concentration gradients. For example, after the sodium-potassium pump establishes a concentration gradient of sodium, then passive diffusion of sodium back into the cell can provide energy for glucose transport. One protein usually moves both the ion and the transported molecule across the membrane. Macromolecules are too large or too charged to pass through the biological membranes, so instead they cross within vesicles. To take up or to secrete macromolecules, cells must use endocytosis or exocytosis. So endo is in, exo is out. So EX like exit, endo like enter. So endocytosis is entering the cell, exocytosis is exiting the cell. Exocytosis moves materials out of the cell in vesicles. So remember, vesicles are just their own little organelle with a bilayer around it that then moves to the bilayer and fuses with it so that it eventually lets what was in it out. The vesicle membrane fuses with the cell membrane and the contents are released into the environment. Exocytosis is important in the secretion of substances made by cells such as digestive enzymes and neurotransmitters. Endocytosis brings macromolecules and particles into the eukaryotic cells. The cell membrane invaginates or folds around the particle and forms a vesicle. The vesicle then separates from the membrane. So it's pretty much the opposite of what happened before. So you have a particle here. So the membrane starts out straight. It senses a particle that it wants there. It moves around it using microtubules and such. It then pinches off. And then that moves into the cell. So it's going into the cell, out, in. So here are more pictures of endocytosis. So you can see how it moves around the particle here and takes it into the cell. And here you can see how the secretory vesicle goes up to the cell membrane and releases the stuff into the environment or the outside of the cell. So endocytosis depends on receptors, proteins that bind to specific molecules called ligands. The receptors are integral membrane proteins on the cell membrane. The resulting vesicle includes both the receptor and its ligand, plus other substances present near the site of invagination. Phagocytosis, or cellular eating, is a specialized cell engulfs a large particle or another cell. A food vesicle, phagosome, forms and usually fuses with a lysosome where the contents are digested. Pinocytosis, or cellular drinking, vesicles are smaller and bring in fluids and dissolved substances. So phagocytosis is kind of like an amoeba eating. It's got this large particle that it takes in. Pinocytosis is more it takes in particles and fluid that are smaller. So receptor endocytosis brings specific large molecules into a cell via specific receptors. This allows cells to control internal processes by controlling location and abundance of each type of receptor on the cell membrane. It also plays a role in cell signaling. So here we have a membrane that's lined with receptors. It can then bind to receptors on the particle, and then it knows that it should take that in. So the receptors are located in membrane regions called coated pits. The cytoplasmic surface of a pit is coated by another protein, often clathrin. When receptors bind to their ligands, the coated pit invaginates and forms a coated vesicle. Clathrin stabilizes the vesicle. So that's what I just drew on the last slide. Here's a picture of it. Here's outside the cell. You can see the little receptors and the ligands in them. And here's the clathrin molecules stabilizing it. It creates a coated pit, it invaginates, and it creates a coated vesicle. This is an electron microscope version of it. You can see the clathrin molecules down here. You can see the ligands and the receptors. Here's the coated pit. It's going to invaginate just like so and go into the cell. Once inside, the vesicle loses its clathrin coat and fuses with a membrane-enclosed compartment called an endosome. Receptors may be recycled to the cell membrane or degraded in a lysosome. This is an important mechanism for controlling the abundance of each kind of receptor on the cell surface. So now we're on to section 5 of chapter 5, cell signaling. So the membrane plays a key role in a cell's response to environmental signals. Cell signaling, cells can process information from their environment. Signals include physical stimuli such as heat or light, and chemicals, which are ligands, those specific chemicals that the receptors bind to. The cell must have a receptor for the signal in order to respond, though. Following receptor activation by a signal transduction pathway is initiated, a sequence of events that leads to a cellular response. So these are a little bit complicated. It's kind of like tracking what a letter goes through to get to its final destination. There's a lot of mixing and sorting that has to go on. So in a multicellular animal, cells are exposed to many chemical signals. They're autocrine signals, paracrine signals, juxtacrine signals, and hormones. So autocrine signals affect the same cells that release them. So these are self-affecting. Paracrine signals diffuse to and affect nearby cells. So affect nearby. Juxtacrine signaling requires direct contact between the signaling and responding cell. So this one is affect next to / connected cell. So this is like through those gap junctions that go from cell to cell. The signals go through those, so they have to be right next to each other. Hormones travel to distant cells, so these go outside of the cell. through another cell into the bloodstream. So these hormones can signal distant cells. Here's the nucleus and whatnot. Okay. So here we have our target cell and our secreting cell. The secreting cell is sending off these ligands through exocytosis. It also has a signal receptor on its surface. So here it is self-affecting, so that is an autocrine signal, but it also affects a nearby cell, which is a paracrine signal. So it can affect ones with receptors, but it cannot affect ones with no receptors. So here, in the case of hormones, the secreting cell secretes it, it goes into the bloodstream, and then it can give the signal to cells that have receptors. So these are the target cells. And it will create a response in them. Only cells with the necessary receptors can respond to a signal. The target cell must be able to sense it and respond to it. It's like you can shout as much as you want behind a deaf person, but they're not going to know you're shouting at them. So a cell without the target receptor is like a deaf person that you're shouting at. They can't respond to it. A signal transduction pathway involves a signal, a receptor, and a response. So here's our signal molecule, here's our receptor, and then there's the response. So there is an inactive signal transduction molecule, which is then activated by the signal molecule receptor complex. It then goes and activates other molecules, which can deactivate enzymes or activate enzymes for short-term responses or cell movement. And then for long-term responses, it goes and affects the DNA expression. It will either go and block a section of DNA so that mRNA cannot be made, or it will go and promote a section of DNA so that mRNA will be made more. We'll go over that a lot in Chapter 11. Signal transduction pathways often include allosteric regulation. So protein shape changes as a result of a molecule binding at a site other than the active site. For example, a ligand-gated channel. A signal transduction pathway may produce short or long-term responses. So allosteric regulation versus competitive. So this one, the ligand binds in the site of the substrate. Allosteric regulation, the ligand is binding at a site other than where the substrate binds. Receptors can be classified by their location. There are intracellular receptors and membrane receptors. Intracellular receptors are located inside of the cell. Their ligands are small or nonpolar and can diffuse across the membrane. Membrane receptors are located on the cell surface, have large or polar ligands that cannot diffuse through the membrane. Membrane receptors. A chemical ligand fits into a 3D site on the receptor protein. The receptor may have a catalytic domain on the cytoplasmic side. The ligand is an allosteric regulator. It exposes the active site on the catalytic domain. So here we have an inactive protein. Now a ligand comes and fits into it and now changes that shape so that it can fit the substrate into it on the inner side of the cell. So outside of the cell, you can see how it fits its three-dimensional shape, and then the proteins go through the membrane and affect the inner side of the cell. Ligand receptor binding is non-covalent and reversible. So it either binds with hydrogen bonding, ionic bonding, or interactions. It does not bind covalently. It can be reversed and go and function again as the protein. So these proteins are not one use. So down here it says R plus L, which is this. This interchange can go back and forth between RL, which is this complex here. So reversible binding is important because cells need to stop responding to a signal after the appropriate response has occurred. Inhibitors or antagonists can bind in place of the normal ligand. Caffeine binds to receptors in the brain, preventing binding of the normal ligands. So normally adenine would bind, but caffeine has a very similar structure, except it has other stuff on the end of it. So they both fit the binding site. So caffeine blocks adenine from binding, preventing the drowsiness. So it prevents the binding of the normal ligand. So ion channel receptors are ligand-gated ion channels. They change shape when a ligand binds. Acetylcholine receptors on skeletal muscle cells bind acetylcholine to open the channel and allow sodium to diffuse into the cell. So you have your channel, and it's all closed up. So then a ligand comes and binds, and this opens up the channel so that sodium can go on through. Protein kinase receptors also change shape when a ligand binds. The new shape exposes or activates a cytoplasmic domain that has protein kinase activity. It modifies proteins by adding phosphate groups. Not all protein kinases are receptors, though. So phosphate groups are just that circular P. They have the P, the O, the O, the O, and the double bonded O. And one of these is negative. So here we have our signal, which is insulin in this case. So this is a major signal transduction pathway in all cells because it tells our body whether to take in glucose or not. So here's the signal insulin. It goes into the receptor, and you have your protein kinase domain, which is inactive. So when it's activated, it has an extra phosphate group added to it. This can now activate a target molecule into an energized version. So the more phosphate groups that you can detach, the more energized you are. So it has a phosphate added to it. It is now energized. So it can then go cause cellular responses inside of the cell. So G protein linked receptors, this is where ligand binding on the surface exposes a site on the cytoplasmic side, which is the inside of the cell, that binds to a mobile membrane protein, a G protein. The G protein is partially inserted in the lipid bilayer and partially exposed on the cytoplasmic surface. Okay. Many G proteins have 3 subunits and can bind 3 different molecules: the receptor, GDP, and GTP, used for energy transfer. So GDP is another one of those molecules like ATP, which is used to store and transfer energy. It just uses guanine instead of adenine. So it looks a bit like this. It's just a guanine here. And then it has the 3 phosphate groups. This is GTP. If it loses one, and looks more like this, this is GTP. It just means guanosine triphosphate and guanosine diphosphate. So this just means tri, and this means di, meaning 2. An effector protein that causes an effect in the cell is also able to bind to it. So the activated G protein link receptor exchanges a GDP nucleotide bound to the G protein for a higher energy GTP. The activated G protein activates the effector protein, leading to signal amplification. So this is like this person goes and tells 2 people something. Those 2 people can then each go and tell a group of people. Or let's do this for spreading messages so that it makes a little more sense. And then each of those can go and tell another group. So you can see how it just gets exponentially amplified. So here we have, outside of the cell, we have our signal hormone or ligand. It binds to the G protein. The G protein linked receptor, well, this is the G protein. This is the G protein linked receptor. You can see how it has the 3 binding sites. It's inactive here because GDP is the lower energy version. So now this changes shape when the ligand is in place, giving its phosphate to the GDP, making it into a GTP. This now activates the G protein, which causes one of its little binding sites to go over and activate the effector protein. It gives up its phosphate to go over here and energize the effector protein, which then can convert reactants into products, which go each creating cellular responses of their own. So a signal activation of a specific receptor leads to a cellular response mediated by a signal transduction pathway. Signaling can initiate a cascade of protein interactions. The initial signal is amplified and distributed to cause different responses, ultimately leading to changes in cell function. There are many ways in which cells respond to environmental signals. Opening of ion channels changes the balance of ion concentrations between the outside and inside of the cell, results in a change of electrical potential across the membrane. So this allows the sodium and potassium to go into equilibrium. Alterations in gene expression. Genes may be switched on, upregulated, or switched off, downregulated, blocked, or promoted. This affects the abundance of proteins, often enzymes, thus changing the cell function. Alteration of enzyme activities, more rapid responses than those involving changes in gene expression. So they can either alter gene expression or the enzymes themselves that are already present. The same signal can lead to different responses in different types of cells. So, for example, the heart and digestive tract muscle cells respond differently to epinephrine because the signal transduction pathways stimulated are different in the two cell types. So, in the heart, you'd want it to beat faster and give you energy. The digestive tract, you want to slow down because you don't want it taking your energy while you're having to have a fight or flight response. So often there is a signal molecule intermediary, a second messenger between the activated receptor and the cascade of responses that ensues. In the fight or flight response, epinephrine or adrenaline activates the liver enzyme glycogen phosphorylase, which catalyzes breakdown of glycogen for quick energy. So you have glycogen stores. This is a carbohydrate that is highly branched. Energy storage in animals. So remember glycogen. It's stored in the liver and in muscles. So it activates the liver enzyme glycogen phosphorylase, which catalyzes the breakdown of glycogen for quick energy. Researchers found that glycogen phosphorylase could be activated by membrane-bound epinephrine in broken cells, as long as all parts were present. They discovered that another molecule delivered the message from the first messenger, epinephrine, to the enzyme. So here's their hypothesis. A second messenger mediates between the receptor activation at the cell membrane and enzyme activation in the cytoplasm. So they have their liver, and they took cells from it and put in each tube. So here on the left, the cytoplasm contains inactive glycogen phosphorylase. And here, the membranes contain epinephrine receptors. So they took the cytoplasmic contents with the glycogen phosphorylase and put it in the one with the epinephrine receptors, and they added that to a secondary tube and then added that to the inactive glycogen phosphorylase. Active glycogen phosphorylase is then present in the cytoplasm. So their conclusion, a soluble second messenger produced by hormone-activated membranes is present in the solution and activates enzymes in the cytoplasm, because they didn't take the membranes out of here, they just took the liquid. So homogenate enzyme activity units, homogenate plus epinephrine had this effect, cytoplasm fraction had very little effect, cytoplasm plus epinephrine still very little effect, membranes plus epinephrine no effect still. So these all are no effect. So cytoplasm plus membranes plus epinephrine, you get an effect again. So these two are the only ones with effects. So they need the cytoplasm, the membranes, and the epinephrine to give a response. So here the homogenate is just the membranes plus epinephrine. So the homogenate is just the liquid solution. So membrane plus epinephrine, the solution created from those two is the homogenate. So the homogenate oh no, that's just the cytoplasm content. I don't know what they mean by homogenate, apparently. But this, the homogenate and plus epinephrine has the cytoplasm, membranes, and epinephrine. So I guess the homogenate is just the membrane plus epinephrine. So it needs all three things to take effect. The second messenger was later discovered to be cyclic AMP, or KIMP. So this is cyclic adenosine monophosphate. So you have your phosphate group, you have your, here it's actually bound twice, which is why they call it cyclic. So this is cyclic adenosine monophosphate, and then it has the adenine over here. Rough sketch there. So second messengers regulate targeted enzymes by binding to them non-covalently. They allow the cell to respond to a signal membrane even with many events inside the cell. They distribute the signal. They amplify the signal by activating more than one enzyme target. So the ATP gets made into CAMP plus 2 phosphates going off. So here is adenine triphosphate, so you can see the 3 phosphate groups there. And here is the cyclic AMP. You can see how it is attached twice, and it gives you that cyclic formation. So that's why 2 of these have to go off by themselves and leave that alone to bind again to the oxygen down here. So signal transduction pathways involve multiple steps in which enzymes are either activated or inhibited by other enzymes. In liver cells, a signal cascade begins when epinephrine stimulates a G protein-mediated protein kinase pathway. So remember you have your receptor with the little tail on it with phosphates on it, which then can activate the G protein, which has a GDP on it, which then can be made into GTP, which is energized, which then goes and activates a enzyme that actually has catalytic function, or can make, it has catalytic function. So then it goes and becomes GDP, and this gets activated, which then can activate other target molecules. Which then go give cellular response. Cyclic AMP is produced and activates protein kinase A, which phosphorylates two other enzymes with opposite effects. So it can either inhibit glycogen synthase is inactivated by phosphorylation, which prevents glucose storage. Activation, phosphorylase kinase is phosphorylated and starts a cascade that results in the liberation of glucose molecules from glycogen. So it inhibits the storage and it liberates the use of glucose. So here we have epinephrine. This is the kinase protein, which then activates the G protein that was there, because there were 3, which then went over here with its activated GTP, which then activates the adenyl cyclase, which then transfers that phosphate group. So now ATP gets turned into cyclic AMP, which then activates the inactive kinase A, which the active kinase A deactivates the glycogen synthase and activates the phosphorylase kinase. So this does 2 things. This does 1 thing, this does 1 thing, and this does 1 thing, and then it does 2 things. So it has an exponential response. So then the active phosphorylase kinase inactivates the glycogen phosphorylase, activates the inactive glycogen phosphorylase. So 1 thing. And then that takes glycogen and turns it into glucose 1-phosphate, which ups the blood glucose for needed energy. The original signal is amplified at every step in the cascade. Each molecule of epinephrine that arrives at the cell membrane ultimately results in 10,000 molecules of blood glucose. Signal transduction ends after the cell responds. Enzymes convert each transducer back to its inactive precursor. The balance between the regulating enzymes and the signal enzymes determines the cell's ultimate response. So a lot of the times at the end, enzymes will be activated, which then go and turn everything up here back to normal. This is usually an end product of the original cascade. So in this case, if blood glucose gets too high, it inactivates the epinephrine receptors. So here, inactive enzyme gets activated by ATP, the active enzyme, then gets inactivated again by protein phosphatase. Here we have GDP and gets activated by GTP. It then gets deactivated by GTPase. If it ends in ase, it's an enzyme. So, and then adenyl cyclase gets turned into CAMP, and then it gets turned into AMP via phosphodiesterase, which then can be added phosphate groups to make ATP. Again, cells can alter the balance of enzymes in two ways: synthesis or breakdown of the enzyme, activation or inhibition of the enzymes by other molecules. Caffeine is a large polar molecule that binds to receptors on nerve cells in the brain. Its structure is similar to adenosine, which binds to receptors after activity or stress and results in drowsiness. Caffeine binds to the same receptor but does not activate it. The result is that the person remains alert because the adenosine, which is just adenine plus a sugar molecule, that's adenosine, but does not activate it. And the person remains alert. So here is caffeine, here is adenosine. It has that 5-carbon ring, and then the 2 rings for the adenine. And then caffeine has its same structure, the same rings as adenine, but it doesn't have the nitrogen group there, and it has a methyl group there, so it doesn't actually activate the receptor. So that is it for chapter 5. I hope you guys learn a lot from watching this and that it helps you. Have a good day, guys.